Hello, everybody. This is Tony Heil with ALS United Mid-Atlantic, and this is our Goals podcast, where we talk about different issues affecting ALS from all sorts of different perspectives, from people living with ALS to caregivers, family members, staff here, like our social workers and nurses, and researchers who are helping to lead us towards new treatments and a cure. As always, if you're interested in learning more about ALS and our work here in Pennsylvania, New Jersey, and Delaware, you can visit alsmidatlantic.org. Our mission is to provide care, advocacy, and research into ALS, and that includes some top-notch researchers from here in our area that we have helped to fund and support to accomplish our mission. And one of those people I'm happy to have as our guest today, her name is Dr. Daniela Zarnescu. She is at Penn State University in the Penn State Health. She received her Doctor of Philosophy from Penn State University in Biochemistry and Molecular Biology in 2000. She also worked at Emory University School of Medicine and spent some time at Arizona University as well. So she has some experience across the country, and including that experience is collaborating with scientists in her own area in Pennsylvania and Penn State and throughout the country to understand things like ALS, how the science in ALS translates to other diseases, and how to successfully complete and participate in different clinical trials. So we're going to learn a lot today. She has so much to say about the progress in science and how you can be empowered to participate in it. So Dr. Zarnescu, thank you for speaking today.
Thank you so much for having me. It's a pleasure to be here and share some of our research interests and accomplishments with your group.
And so first of all, I gave a little bit of a background of your education, really, but there's so many different fields you can go into in science and medicine. Was there anything that made you really focus on things like genetics and then ALS? Was there something that spurred your interest in that?
Oh, that's a great question. So when I was an undergraduate student at University of Bucharest in Romania, I studied physics. And as much as I love physics and the quantitative aspects of physics and math that comes with physics, I had an opportunity to move to the United States, and I heard a scientist talk about genetics, and I became fascinated with the possibilities in the field of genetics, and I thought that's a perfect area to apply my knowledge of and my quantitative skills to a new area of science. And so I went to graduate school at Penn State in the Department of Biochemistry, learning molecular biology and studying genetics.
And we have had some great researchers from around the world who have participated in ALS research, whether it is through universities or drug companies from, like you said, Romania, Europe, Asia, all over the world. How important is it for America to be recruiting some of those minds to come here to collaborate on science?
Well, Tony, America is still the best place to do research. I think the investment in science that the US government has made, as well as foundations such as yours, as well as companies, has really made this the best place to do scientific research and be able to push the boundaries of knowledge and scientific discovery. And I hope this remains that way.
And Penn State is nationally and globally recognized as one of the top research centers for a wide host of topics, especially ALS, and we're proud to work with them. What is that process like at Penn State Health? Like, how do you work together on science? Is it just you and your team, or do you work with others as well?
Well, science is very interdisciplinary, and today, to really make an impact, especially in the lives of patients, one needs to take an interdisciplinary approach. So we cannot do effective science in isolation. And so I have many collaborators that can help us answer our questions using different approaches, using their expertise towards the ultimate goal, which is to better understand the mechanisms underlying amyotrophic lateral sclerosis and develop therapies. So we have a terrific collaboration with the clinic here. The ALS clinic at Penn State is very strong, is led by Dr. Zach Simmons, who is a close collaborator. And at the clinic, there are also other clinicians, such as Dr. Jim Grogan, as well as Bill Shu, And both of them actually have been interacting closely with my graduate students and my postdocs, and have given opportunities to my students to observe them in the clinic, meet patients, and really understand the challenges that patients face, to really bring it back to the lab and help us better understand and better formulate the questions that are important to study that can have an impact in a patient's quality of care and life.
And we have worked with
There are
Dr.
other,
Simmons
sorry,
for
there are other
almost.
basic scientists as well, I should say,
Mhm.
but the collaboration with the clinic is really important. And that was actually one of the drivers that made me move to Penn State. The strength of the clinic, then the ability to collaborate with clinicians and have the ability to more directly impact the patient's lives.
I appreciate that. We've worked with Dr. Simmons for nearly 30 years now as I record this. He received an award last year from the International Association of ALS MND Associations. And, how important is it to have a leader like Dr. Simmons that brings in people like the other doctors you've mentioned, the post grad students? I know he has a great reputation that makes people want to learn from him, right?
Yeah, very important. Like I said, talking to him during my interview here was a major factor in my selecting to come to the College of Medicine at Penn State. He has a reputation of being a terrific clinician as well as a very kind individual. And his generosity in, sharing the time that he has, both him as well as the other clinicians in the ALS clinic, to share with my students and with us what are the most important challenges from a clinical perspective. So then we can come back to the lab and really design the best experiments and take the best approaches to really try to make a difference in ALS research.
And you've had research funded in very different ways. There's lots of different ways that you can receive funding, whether it is through some grants from us at ALS United Mid Atlantic, the Congressionally Directed Medical Research Program. So let's talk about that, the CDMRP. I know that you had a study that was funded through that. What kind of study was that? What did you do?
Yeah. CDMRP funding, specifically for ALS, is critical for making progress in this area. Obviously, it is of interest to the Department of Defense
Mhm.
because veterans are at higher risk for developing ALS. And I have been funded by the Department of Defense through this program more recently to better understand and develop small molecules with neuroprotective potential that have the ability to be developed into drugs for treating ALS. So more specifically, when I was at the University of Arizona, I had a collaboration with a colleague in the chemistry department. And we got together and developed a project whereby we would take some of the small molecule that he's been synthesizing in his group, and we would test them in our models of ALS that we developed in my lab. And we call this approach phenotypic screening, meaning we developed ALS models in Drosophila that exhibit survival deficits as well as locomotor deficits, two things that are also present in ALS patients. And what we did is we treated our models with these novel compounds, and we found that they can increase the survival and improve the locomotor function. We then proposed to the DoD to better understand how these small molecules are working. How are they really working in the neuron to improve the function and the survival of the neuron and of the whole organism? And as part of this project, we also added a collaboration with a company called AcuraStem, which is a leading biotechnology company in California, funded by a collaborator of mine, Dr. Justin Ichida at University of Southern California. And the reason we added this collaboration, again, bringing back that need for interdisciplinarity, is because this company has developed a platform for screening compounds and potentially new drugs with neuroprotective potential in ALS in neurons derived from patient stem cells. It's a technology called induced pluripotent stem cell technology, whereby cells are taken from patient skin and differentiated in a dish to become neurons. And so what we did is we tested our top compounds from our Drosophila models of ALS into patient neurons and found that also in patient neurons, these compounds can increase the survival of these neurons, like I said, derived from patients. So this is a powerful approach that we've been taken and has been funded by the CDMRP program, where we do the discovery work in our simple models, which are fast and relatively less expensive than rodent models, for example. And then we take it directly to human neurons, patient-derived neurons, to identify those that have protective potential. We have a patent on these compounds, and Accurastem is interested in licensing our technology as we make progress through this. So we're happy to say that we have a better idea now of how these compounds work and how they might be used to be developed into drugs that can treat or improve the quality of life of patients with ALS. And I want to add that having this funding from the Department of Defense helped us make progress. So last year, my collaborator from University of Arizona and I got funding from a foundation called the Critical Path Institute, which is kind of a public-private enterprise. And they are funding us to understand the physical, chemical properties and the toxicity of these compounds to help us further push them into drug development pipeline. And hopefully one day they could be developed into therapeutics for ALS.
That's incredible, both the science and the cross-country collaboration with at least 3 states there. Now, when you're doing the screening, whether it's you or the company, what exactly are you screening for? Is there something in particular you're looking for as a number or something in particular?
Yeah, we're looking for one of the simplest effects that we can have both in vivo in our models as well as in vitro in the patient-derived neurons, which is improvement in survival. Neurons from patients with ALS that harbor mutations that cause ALS or derived from patients with sporadic ALS don't live as long in an animal or in a culture dish as a normal neuron does. And so we take that difference in their ability to survive, and we use that as what we call a phenotypic readout. So we're looking for the ability of our compounds to increase the survival of these neurons. And then once the compounds pass this initial test, we can go a little deeper and ask, are these compounds improving the synaptic morphology and function at the neuromuscular synapse, for example, which is altered in ALS patients? Are we reducing other biological effects associated with ALS, such as oxidative stress, the presence of aggregates, and other metrics? So one by one, we are testing them for their ability to make improvements in these processes that we know are altered in ALS patients.
And you've been working in these fields for 25 plus years now, really. So, are the things you're looking for now different in some way because you have a new knowledge? You and all of science have a new knowledge of ALS and genetics than we did 25 years ago. So, you have new things to look for today?
Yeah, I've been working on ALS for only 18 years, but it's still a long time. And that's
Only
because
18 years.
only 18 years. And that's because while science is fun and exciting, it also takes a long time if you want to do it right.
Mhm.
But I would say it's been exponentially increased. Our knowledge about ALS, the genetic basis of ALS, the connection of sporadic ALS, the understanding of the cellular pathways that are altered in a degenerating neuron, what other cell types are involved, has really grown exponentially in the last 18 years or so. And that has been driven in part by this interdisciplinarity and by the ability of scientists to collaborate across disciplines, across institutions, across the country, across the world, that really brings together the best people that can answer a specific question with the right tools. So I would say the landscape has changed dramatically and continues to change. It's quite fast-paced right now, and I think it's a really exciting time to be doing research in this area.
Well, one of the things you've been researching has been environmental connections with ALS, and that's something that ALS United Mid-Atlantic has been proud to help support and fund in the last year. So, what exactly did we fund, and how are you working on that?
Well, first of all, thank you for funding us. It's a really exciting collaboration with the clinic that have been thinking about for a long time. We do know that environmental factors, whether they're chemicals or particulate matter in the air, have a contribution to ALS. And there's been good progress made by a group at the University of Michigan, as well as sporadic other places around the country. And while we knew that perhaps a significant contributor to sporadic ALS is our environment, we don't really understand how that plays a role in the development or onset of ALS or the progression of ALS. So I thought the collaboration with the clinic was a perfect opportunity. And we now have a project in place where the clinic can start enrolling patients. So if you're listening, please consider the call to enroll in this study, where we are first administering a survey to better understand the potential exposure to environmental toxins that patients have had throughout their life, either occupational, depending on the professions that they had in their life, or residential exposure, depending on where in the country or where in the state they lived. And we have a collaboration, in addition to the clinic, we have included a collaborator from the Public Health Sciences Department at Penn State that gives us the ability to take that zip code and use something called geocoding that geographers use in a very different field to basically be able to tie in that residential information to potential exposures to environmental factors in our living situation. So we will be getting that information from the survey. We will also be asking the patients to kindly give us a small amount of blood to do two things. One is we will measure the presence of specific metals, like heavy metals that are thought to have potentially a detrimental effect on neurons. And then also we will use some of that blood donation to do a genetic analysis. It's an analysis called SNP analysis, where we will look for specific modifications in the genome of these individuals. And then using sophisticated statistical analysis, we will combine the information that we get from the survey and from the blood, both in terms of metal presence as well as genetic changes that together could constitute risk for developing ALS. And then we'll take that information and we'll probe it, or we'll test it in our simple Drosophila models, and where we will administer some of those risk factors, whether it's a metal like lead or arsenic that's in the environment, and administer to our Drosophila models, and then measure the effect of these exposures to locomotor function, survival, potential formation of aggregates that could explain the risk that these factors are indicating to us from the human study. So we're going to go back and forth between human information as well as Drosophila modeling, both in terms of environmental exposure as well as genetic information, and try to better understand risk that will help us come up potentially with prevention approaches, but also understand how current exposure to these metals could contribute to progression of disease, whether it accelerates or mitigates the progression of ALS.
Is that something where, when you understand some of those environmental issues, you could talk to people who may be more susceptible to ALS, like it's in their family. They may have the gene, and now in the future, we know, okay, you should definitely not do X, Y, or Z, or you should be cautious of being in this kind of field because you could be exposed. I don't want you to call anything out specifically, but is that something in the future that we could consider?
That's the goal. We would like to be able to say X, Y, and Z factors contribute to risk or accelerate progression of disease. Therefore, a recommendation could be put in place to avoid, like you said, these factors, or potentially we can see how these factors are affecting the function of neurons and come up with a therapeutic strategy, very much like a drug therapy, for example.
Mhm.
So both are possibilities, but it's exciting to think about the possibility of prevention
Mhm.
in addition to the possibility of intervening and mitigating the consequences of disease. So we would love to be able to do that.
Yeah, and I've been talking with some other researchers about being able to diagnose more quickly because understanding biomarkers and things along that line. So, if you combine those fields together where you can kind of diagnose faster and also know some ways to prevent, maybe you could diagnose much sooner because you can see certain indications in a person's blood, iron levels. I'm not the expert, you are. Uh, and then you can contribute, you can add to that the information you have with, okay, you're early on, so we can tell you a better therapy plan.
Yeah, that's a very good way to think about it. And I think this will leverage information that comes from different fields. And actually now, many of us are thinking about how we can use artificial intelligence to combine the information that we get from all of these types of studies, whether it's diagnostics or biomarkers or information about prevention. I think you'll see in the next years our ability to analyze these large data sets, large amounts of information to potentially synergize together and help diagnostic, prevention, treatment. I foresee all of these being possible in the next few years.
And one other thing from the last few years has been that collaboration again. I want to bring that up because you mentioned Michigan as the fourth or fifth state you said already today. Is
Mhm.
it much easier to collaborate now than when you first started in these fields because you have Zoom, Teams, all sorts of things online where you can share information? So is that collaboration better because of the technology we have today?
I think the technology helps, but I also would say I've worked in other areas of research, and I think the researchers that study ALS tend to be collaborative and tend to share information, which is really a privilege to have, actually, to be able to collaborate and share data and tools and reagents. Um, I think it's hugely important. There's something about ALS researchers that have that. But you're right, having the ability to jump on a Zoom call and share data or ask a question or get advice from another expert is really helpful. It's a little easier than jumping on a plane and making it to Arizona, for example. I still have many collaborations back in Arizona, both at the university as well as the Barrow Neurological Institute, as well as new collaborations here in Pennsylvania at Jefferson and Pittsburgh. So it's a really exciting opportunity to push the field forward.
Yeah, my grandfather had ALS and he passed away in 2007. And his brain tissue is with his doctor in Pittsburgh, Dr. Lacomas. And I know that the tissue
Mhm.
he donated is still contributing to studies that could benefit your work. So a person with ALS can leave a legacy when they participate in any of these studies and donate their tissue and blood, right?
They sure do. And that's actually really, really important. And we very much appreciate, myself and others do a lot of our studies in experimental models, and this is great. But ultimately, we need to be sure that our discoveries and our work is directly applicable to patients. So having access to that donated material, for which I thank you, it is really, really important to be able to validate our research findings and determine the relevance in the patient population.
And I've talked with a number of patients who participate in research. Some of them have been involved with our committees to fund research. Dr. Stacy Lewin-Farber and John Russo, they've been very involved in this. And they say that research is so empowering. You talk about the work that you have with a clinic. Not every researcher has that benefit, obviously, but how important is that clinic to empowering patients to be involved in research?
Well, I think they do a great job sharing the possibilities that can, or the outcome of getting involved in research. I would love to be able to do more of that because I think it's mutually beneficial. It's beneficial for us who sit in the lab and do experiments to really understand the potential impact that our work can have. And I think it can only push the discoveries further if we are able to communicate to patients the work that we do, the dedication that we have in the lab, um, and the passion for really trying to figure out therapies that can be helpful to them. And I also want to add here that one of the aspects that maybe people don't think about not being in a research lab is the pay forward work that we do by training the next generation of scientists. Um, the students and the postdoctoral fellows and the lab personnel are the next generation of researchers in ALS. So giving them the opportunity to go to the clinic and shadow the clinician and meet the patients and vice versa. I think if the patients see the dedication and the passion in these young individuals that will take the field forward, and literally it's workforce development if you want to think about it that way. It's really, really important. So I hope more opportunities are available for us to interact in this way and really understand the importance of each player in this area.
And I know a number of the people from the Penn State Health Hershey Clinic, whether it's the doctors, nurses, or researchers, will be at our ALS United Hershey Walk, which is great. We love to see everyone there at that walk. So please go to our website at alsmidatlantic.org to learn more details on that as well. And just one other thing I'd like to bring up, because we talked about the collaboration with you and the other people across the country in your own clinic, and some of the things you've learned that may be attributed to applicable to other diseases. So I mentioned my grandfather, he had ALS. My grandmother on the other side of my family had Parkinson's disease. You and I have worked on some legislation and some ideas for brain research in general. So are there things that either that people have learned from ALS that could help other diseases, or vice versa, where you look at that and you say, oh, Alzheimer's is doing that. We should kind of go down that street a little bit, or, oh, Parkinson's is doing that, but maybe we shouldn't because that shows that this wouldn't be as helpful here.
Absolutely. First, let me tell you that I would love for us to participate in that Hershey Walk as well, my whole lab. But when it comes to your question about are we learning from each other, very much so. And I just want to make a comparison to the cancer field, right? We have made so much advancements in cancer research. Of course, it's a little easier to take a tumor and study it, much easier than taking neurons and studying them in a dish. But the investment and the ability to look at cancer holistically, at what different types of cancer share, I think it really helped the field move forward and make many cancers a chronic disease. Recent advancements in ALS, Parkinson, Alzheimer's, traumatic brain injury, and other neurodegenerative disorders have really highlighted the fact there are actually 7 common cellular pathways called hallmarks of neurodegeneration that have recently been reviewed and highlighted as common processes that are altered in all of these diseases. It's really remarkable if you think about it, because a protein like TDP43, which is mostly linked to ALS, but now linked to frontotemporal dementia, it's a protein that's expressed in the whole body. Every cell in our body expresses TDP43. Yet, when there is a problem or a mutation, motor neurons are the ones that suffer first. However, the way those motor neurons are suffering is very similar to the suffering of the dopaminergic neurons that are affected in Parkinson's disease, where there is another protein, alpha-synuclein, that is mutated or is causing problems. Similar processes are altered. For example, the way cargo moves in a neuron from the nucleus to the terminus to the synapse, the way the neuron deals with stress, including oxidative stress that can be caused by injury or mutation or exposure to environmental toxins. The way neurons die, it's very similar in Parkinson and Alzheimer's and in ALS. The way those synapses, those places of communication between neurons or between the neuron and the muscle in ALS, are all going down or going wrong, if you want, in very similar ways. Yes, we have a lot to learn from each other, and I think it's really a perspective changer. And I'm hoping that this more holistic approach can help us make more progress and learn from each other that studied the different diseases. For example, now we're also interested in learning about frontotemporal dementia, exactly for the reason I mentioned. TDP-43, which is our major focus, forms aggregates not just in ALS patients, but also in frontotemporal dementia patients. These protein aggregates, the way they form and the way they are toxic, and strategies to get rid of them to improve neuronal function are probably much more similar in these different diseases than we previously thought.
Wow. I feel like I deserve some continuing education credits for just that answer alone. Uh, my grandmother, when she had Parkinson's, she had a deep brain stimulation surgery, and it was remarkable. My mom was in the surgery room with her and saw her go from great shaking to just as if she didn't have Parkinson's at all. But her disease had progressed so quickly. I mean, it's so much at that point that it could only do so much. If she had been able to get that sooner, it would have been a big effect. And I see that Penn State Health is a leader in that as well right now. So
Yeah,
it probably makes a big difference that a place like Penn State Health is a leader in Parkinson's and ALS because you guys can work together. And so anyone who's interested in one, well, you can just go down the hall.
absolutely. And actually, more and more of these interactions are happening between us in the basic science departments and physicians and clinicians in the Department of Neurology. We were just having a conversation last week about what potentially from our work on ALS and frontotemporal dementia could be relevant to studying Alzheimer's. So all of this is happening, and I think it will really help us better develop treatments for these different diseases, this holistic approach.
That's incredible. And I could end it here, but I have to ask because you brought it up about stress. I've talked to a number of people just since I've been doing this podcast here about the very emerging discussions on stress. You understand that from a scientific background, but as I've talked with some people, there's also a demystifying stress, and because people didn't talk about it enough, and they thought it was just something you needed to just take a nap. And now we understand so much more. How important is that research and understanding of stress to disease in general?
So important. Thank you for bringing this up. There are many ideas that are trying to connect stress to the development of neurodegenerative diseases at the molecular level. And I just want to share a quick cell biology fact that it's very exciting and easy to understand. When a cell is exposed to a stressful condition, whether it's environmental or some kind of injury or a presence of a mutation or maybe aging, that stress literally undergoes, that cell literally undergoes a physical remodeling, and molecules inside the cell forms protein complexes called literally stress granules. These stress granules contain many of the RNA binding proteins that are linked to ALS, like TDP-43, G3BP, and others. I'm not going to name them here, but there is an intimate molecular connection between presence of stress around the cell and the cellular response in the form of protein accumulation in these stress granules. They also contain RNA. And without going into a molecular biology lesson here, I would just say when that happens, those RNAs cannot be made into protein like they normally would. So there is immediate direct effect on the gene expression program of that cell. And some of the ideas are that if the stress doesn't resolve quickly, these molecular um, structures, stress granules persist, and they can accumulate and cause additional problems like generation of free radicals that can damage the cell, and a continuous detrimental effect on gene expression. So there is an actual physical response that we can visualize under the microscope called stress granule formation that is directly tied to stress. And we and others are interested in understanding the connection between this molecular response, aging, and how that may tie in with all of these neurodegenerative disorders, including ALS, that ultimately have as the biggest risk factor age.
Wow, that makes a lot of sense. And it also shows why, as you're aging, to be considering that, and you can understand different kind of whether it's drug therapies or occupational therapies or physical therapy, whatever it is. I'm not the doctor, but I think that there are a lot of great things happening to understand that, as you said, especially as you age. Really appreciate it. Dr. Zarnescu, thank you so much for speaking today and sharing your information. Now, if people are interested in learning more, they don't have to be a patient at the Hershey Clinic to participate in your studies, right? They can. How can they participate in any of these studies?
I believe the clinic has now an announcement on their website of how you can enroll. But I'm happy to share that with you by email, and perhaps there's something you can post on the, would that be a possibility to?
100%. We'll put it in the description here when
Yeah.
this is live on our YouTube channel and on our
Mhm.
website so people can see that, and in the description of the podcast so people who are listening can go and learn more about how to contact Dr. Zarnescu, Dr. Simmons, and the whole team. And of course, if you're interested in research in general, go to our website, alsmidatlantic.org. And just like Dr. Zarnescu, we partner with people from all across the country because our goal is finding treatments and therapies to end ALS and to make living with ALS much better and less stressful for everybody, including caregivers as well. So, Dr. Zarnescu, thank you so much for speaking today.
Thank you so much for having me, and I look forward to continuing interaction with your group and your
Great.
patient. Thank you.
It is our goal to end ALS. Thank you to everyone who is involved in our mission.
Thank you.